you guys. So I was up here a little bit ago more in my parent capacity, and now I'm up here more so in my researcher capacity. So I'm Miranda McManus. I have a 19-year-old son with NF1, but I'm also on the biology faculty at the College of Charleston and teach genetics at the college level. And gene therapy is not something that is available for people with NF yet. But you may have heard there are a number of gene therapies that are actually out there on the market now. The past several years have really brought this into the forefront a little bit more. And we've got a ways to go for NF, because NF is complicated in a lot of different ways, but it is on the horizon, and it's kind of a complicated topic. And so it's really important that the community starts to prep for this, right? That we start to educate ourselves and understand what patients want in gene therapy, so that the first gene therapy clinical trials are successful, because when they do start, they're going to be small. So we need to have the right patients involved and need to be addressing the right things. So before that, though, comes a little bit of education. And so that's kind of where I'm going to go today. And so it's not working for me either. Yeah, it is not just you. Let's see if it works now. All right. So if it happens to anybody else, touch the slide, then it'll work. All right, so first, little crash course in genetics. Now, I take a semester to teach this to undergraduate students that have had a ton of background already in biology. And if they're going to be geneticists, they go on to take a whole lot more. So in this, few minutes that I have, there's no way that we're going to be able to cover everything. So I'm just going to try to touch on some of the basics and hopefully help you understand how gene therapy can help one day. So when we are talking about gene therapy, it's obviously going to be related to the human genome. Well, if you notice on the first slide, I used the word gene-targeted therapy, and that's because gene therapy is not always involving the DNA. There's actually several different stages before it becomes functional, and it can happen at any point in that process. So in our cells, almost every cell in our body, we have a whole copy of our human genome, right? That's 23 pairs of chromosomes. And if you think of the human genome as being like an instruction manual for everything that happens in your body, each of those chromosomes are kind of like chapters, right? We have 23 pair of them. Of those 23 pair, one pair is a pair of sex chromosomes. That's the last one. And NF1 affects chromosome 17. NF2 and the schwannomatoses affect NF2-related schwannomatosis, and the other schwannomatoses affect chromosome 22. So we're really just looking at a small portion of the genome that is impacted by NF. And so it's part of these 23 chromosomes. And these 23 chromosomes, if you unwind them, then we get these letters that actually build up the whole thing. So A, T, C, and G are the code that makes up this instruction manual. And in this code, there's about 3 billion base pairs, so 3 billion pairs of those letters. And about 20,000 genes. So the genes you can think of as like the words that make up those chapters, right? And so when we're talking about gene therapy, we're kind of focusing on just a region of a particular chromosome, right? So this is a diagram that's showing how NF1 is inherited, but NF2 and the schwannomatoses are inherited the same way. It's called an autosomal dominant inheritance pattern. What that means is autosomal means it's not on the sex chromosomes, right? So we're talking about chromosome 17 or 22 here, 17 in the case of NF1. And for each of us, we have a copy of each of those chromosomes. We have two copies, pairs, right? Because one comes from our mom and one comes from our dad. And in NF, we're only going to see one of those chromosomes being impacted at what we call the germline level. So that is what you can pass down to your children, right? NF is not always inherited. We know that a lot of times it can be spontaneous, but once you have it, it can be passed down. And so this is showing how that happens. So up here, we've got our affected parent that has NF1. So this chromosome is the one that's impacted. If they mate with an unaffected parent, then we end up getting about half of the kids being impacted by NF1, because our affected parent over here is the one that's determining that, right? Half the time they're going to pass down the gene that has the mutation, or pathogenic variant is the word that you'll more commonly hear now, and half the time they won't. So that doesn't mean that if you have 4 kids, 2 are going to have it, 2 of them aren't, right? It's all a probability. It's like a flip of a coin, every child that you have. But about half the kids are going to be affected, and this is with NF1 or the schwannomatoses. So this is where, if we're going to talk about gene therapy, though, we have to go a little bit more in depth, because gene therapy is not just going to be impacting DNA, because DNA has several steps before it gets to the functional protein that actually plays a role in our body. So in NF1, that protein is neurofibromin. In NF2, for example, we have myelin. And so along the way, there's a few steps that happen that get it into this functional protein form. So up at the top, this is our gene. So this is just taking the segment of the DNA. And you can see that there's different parts here labeled exon and intron. And then we have a promoter region that just kind of gets everything started with this process. All of these exons are the coding region of the DNA. So these are the parts of this code that are going to be in the protein, right? That part of the language is going to be in the protein. So in the first step, we call this transcription. Our DNA sits in the nucleus all tucked away in our cell, right in the middle of our cell. It never leaves. But when it's about to be used, a copy is made in the same language. It's RNA. Instead of those letters A, T, C, and G, we're going to be having A, U, C, and G, but it's essentially the same language. It gets copied over into this little segment of RNA. And then it goes through another step, and you guys have probably heard of messenger RNA before now because of the COVID vaccines. Messenger RNA is the part that will leave the nucleus and go out into the rest of the cell to work. And you notice that in our messenger RNA, all of these intron regions that are the non-coding DNA are snipped out, right? So they don't end up in our final messenger RNA. So that is transcription and then this processing step. And then we go through another step that's called translation. This is changing that DNA, RNA language into a completely different language, right? The language of proteins. So proteins aren't made up of those letters, those nucleotides, A, T, C, and G. Proteins are made up of amino acids, and amino acids are coded from that DNA code. So through the translation step, we get this amino acid chain that we call a polypeptide. Even still, this isn't functional, right? Even still, we just have this kind of sprawled out polypeptide that isn't able to do its normal work. So it has to go through some modification after translation where it often folds, sometimes it braids. There's all kinds of different things that can happen to protein that fold it in a way to where it becomes functional in the body, all right? So the thing about gene therapy is that it can really impact any of these different levels, right? We can have the DNA be impacted. We can have this messenger RNA be impacted later on, or the RNA step before that. We can have our protein being affected. We can have the folding being affected, right? So all of this along the way could be involved in what we're actually calling gene-targeted therapy, all right? So just to give you an idea of what some of these NF-related proteins do, well, they tend to be involved in a lot. We know a lot more about neurofibromin than we know about myelin. We don't know the role of some of the proteins completely, but we do understand a lot of the roles. And they can do things like prevent our nerve sheath cells. Remember, Dr. Gross this morning was talking about the cells that are around the nerve cells. It can prevent them from multiplying too much. We call them a tumor suppressor gene for that reason. They can suppress the growth of tumors. They also play a role in nerve cell signaling and in doing things like controlling genes that are related to inflammation, and one thing that we come back to a lot in NF and schwannomatosis, pain. So we're going to talk about a little bit about the different types of genetic variants. And the thing about NF is we're dealing with some big genes that are complex. They have a lot going on. And there's lots of different types of genetic variants that can impact these genes. So it's easier to think about this in sentences since we don't have the genetics background probably in the room. So if you remember, we read this DNA sequence, to get it into our amino acid chain sequence, it reads in letters of 3, right? And so there's 3 letters that end up corresponding to a particular amino acid. Now, we only have 4 letters in the alphabet with DNA, but so that we've got some sentences we understand, I used a few more. But these are all 3-letter words in our sentences. So if our original or our wild type NF1 gene, say, we had the sentence, the red cat ran out the car, we could have something called a missense mutation that just changes one of those letters somewhere. And in reality, sometimes those missense mutations don't even really change anything because it ends up substituting out for the same amino acid. But sometimes it'll change the meaning a little bit, right? And so you might get the red cat ran out the bar. Sometimes that can be a problematic change. Sometimes it's not, right? It kind of depends on where it is. We can also have something like an insertion. So this first insertion, the big red cat ran out the car, is what we call an in-frame insertion. In-frame insertions or deletions, because the next one is an in-frame deletion, just the cat ran out the car, are either going to add or remove 3 different letters, right? But the good thing here is the reading frame remains the same, right? So it didn't mess up the words. You can still read the sentence. You can still make some sense out of it. Some words might be more important than others. Some amino acids are more important than others. Some impact that later folding. Some don't, right? But then we can also get situations where we have what we call a frame shift insertion or deletion. And with a frame shift insertion or deletion, you can see in this case it was just the letter B being added. Then when that letter B was added, it messed up the rest of the sentence, right? So none of the other amino acids in that chain are going to be the same. That can be pretty devastating to a protein product, right? When you have something that just changes all the way through. The same type of thing happens with the last mutation here, a nonsense mutation. A nonsense mutation is when the letters that code for stop, for ending the whole translation process, it's when those letters end up turning into a stop codon, and then in this case it just cut it off after the cat, right? So these can be pretty devastating variants as well, because particularly if this happens earlier on in a sequence, it's going to mess up the rest of the gene, right, in the way that it's read. So those can be much more impactful mutations, but any of them can have a wide variety of manifestations. So those are just if we're talking about a few letters here and there that are being deleted, but those aren't the only types of mutations that we can see. There's also mutations that impact much larger areas of the genome. In some cases, we might see the deletion, say, of even the entire NF1 gene, plus some, right? And so on this slide, the original sequences, which you'll hear referred to as wild type, normal, which is a weird word to use, or functional. You can see that we've got A, B, C, D, E, F, and G here. Sometimes we have duplication mutations, and if you say like A, B, C, D, E, F, and G are actually whole genes, right? You might end up with a couple extra genes on your chromosome for that particular type of gene, or sometimes they can be deleted like we see here. Sometimes they even get up and move around. They flip around. It can be an inversion. And sometimes we'll even see it jump from one chromosome completely to another. So there are a lot of different types of pathogenic variants, or variants in general. They aren't always pathogenic, which means causing disease, that we can have, all right? So lots of different variants. And that's one thing that really complicates gene therapy approaches when we've got a condition like NF1 or the schwannomatoses, that people have very different variants from one another, right? It's not like some other diseases that we see that may just have one single change, and that's what impacts everybody that has the condition. With NF1 and the schwannomatosis, it's not going to be the same. So just to give you an idea of how you'll see some of these variants listed, this looks like a bunch of mumbo jumbo, right? But it's nomenclature that's used to describe these different types of mutations. So if you guys have your genetic report, you might see something that looks like one of these. So this is the DNA sequence. And so like this is a nonsense mutation here. In this case, it changed a C to T, but that ultimately ended up coding for one of those stop codons. And so when we write the protein sequence out, we put this TER that means termination, or this star, this is the same type of thing. We'll call that a truncating mutation. So that just cuts it short, right? That nonsense type of mutation. Frame shift deletions happen, right? And so that's what that FS means in our protein sequence here. And it actually tells you where it happens in some of the sequences. In this case, it's a splice site mutation. Remember, we can have those exons added or removed. And I want you to realize that some of these types of variants, you can actually have two at once. My son, for example, has a missense variant that causes an exon to be skipped, right? So sometimes it's a little bit of a combination of these. Missense, we just see a letter being changed here. In this case, it changes an amino acid. Sometimes that's not going to be the case. And then we can have larger deletions. So again, not really important to understand this nomenclature, but those letters on those genetic reports do mean something. They actually give you a little bit of information, not just about the type of mutation that it has, but often the impact that it has as well. So to talk about how gene therapy comes into this all, let's talk a little bit more about how these mutations get passed down through this whole process, right? So we start off here with our DNA, and we're just looking at a little segment here, right? So that just say we're talking about an NF gene, whichever NF gene you want to think about, it doesn't really matter because they all are passed down the same way, right? They're all autosomal dominant. And germline mutations, you're only going to have one copy, right? For germline mutations, you don't see instances where both of the chromosomes have the mutation on it. So when there is an NF mutation, say right there, it's just SOM1. And when it goes through the transcription process, that mutation also ends up in the RNA. And so when that goes through the translation process, that also ends up impacting the protein. You could end up having no protein. You could end up having a non-functional protein. In some cases, it doesn't have any impact on the protein, right? But you notice here that you still have one copy left, right? And that's the reason that this isn't something that is fatal before birth. With two copies, it will be, right? Because you need to have those tumor suppressor genes or you're going to have an overgrowth of cells. So with just this one mutation, which is your germline mutation, there are some manifestations that are related to that. So things that we see in NF1, like developmental delays, learning disabilities, things like scoliosis or vascular disease, these can happen just with this single variant on the one chromosome. In NF2, cataracts, chronic pain is also caused by just having one of these impacted. But there's a lot of other manifestations that don't happen until there's actually a secondary mutation. So our other chromosome can mutate in the same gene, not in the same place, right? It's just going to be somewhere else in the gene. And when that happens, in NF1 and NF2, we'll see this occur in the same gene, but in LZTR1 and SMARCB1, we'll actually, which are the different forms of schwannomatosis that are not NF2 related, we'll actually see mutations in other nearby regions too. For example, in LZTR1 and in SMARCB1, the NF2 gene ends up getting a hit on both of those chromosomes when we have some other types of issues, right? So that also leads us to think that maybe looking at the NF2 gene with gene therapy might actually be helpful, because perhaps for some of these things that occur later on, because this gets passed down into our RNA and then also into our protein. And that's when we're going to see the tumors develop, is when you have that secondary mutation. So things like neurofibromas, schwannomas, meningiomas, low-grade gliomas are all going to have this somatic, which means not in your germline, wasn't passed down, isn't going to be passed down, right? This is in other cells in your body, that it takes that second hit to develop these tumors, right? And like I said, in the schwannomatoses that aren't NF2 related, it takes 2 hits in NF2 to see those. So we'll see those benign, non-cancerous tumors, also cafe au lait spots, hyperpigmentation, the freckling, all that. If you look at those specific cells in a cafe au lait spot, they have a secondary somatic mutation. So that's the non-cancerous tumors. We actually see and have identified other gene hits that can happen that cause things like MPNSTs or high-grade gliomas, right? So these tend to be in other genes. So these first two here, SUS12 and TP53, are both on chromosome 17, the same one as NF1. And then CDKN2A is on another one. But we see these mutations in cancerous tumors, right? So you can see a lot of different places here that you could potentially target to try to fix things, right? One thing I want you to realize is that we are not doing not even thinking about gene therapy at the germline. So not anything that would be able to be passed down. That would be a long way down the road, right? So it's not going to be, any gene therapy is not going to be a completely curative treatment that takes the NF1 mutation out of all the cells of your body, or that takes your NF2 mutation out of all the cells of your body. But it could have impacts for the future, right? So there's different types of gene therapies that address each of these different stages. So we've got gene replacement. So gene replacement very often uses viruses, which are pretty good at getting into our cells, because that's what they're made to do essentially, but taking those viruses and adapting them to our own means. If we can introduce a gene into certain cells of the body to where it injects itself into the genome, that could essentially replace the gene. And again, we wouldn't be able to do this in every cell of the body, right? This wouldn't be impacting the germline, but this could be enough to make enough neurofibromin, say, for NF1, that it ends up having a positive impact. We can also see gene editing at this stage. So most of you have probably heard of CRISPR before. That's the one that gets all the news. CRISPR for NF1 would be very challenging. It's a really big gene with a ton of different mutations. And so you have to think that you would have a specific, gene therapy for every single type of mutation to be able to replace that, which would be hard, right? And we have similar challenges with the schwannomatoses. So there's some gene therapy work, some gene replacement initial study going on now. There's also some gene editing study. But I would definitely say that for this population, that CRISPR is probably a little bit further down the road. There is, however, an approved one for sickle cell, so it is being used for some things now. We also see at the RNA level, you can have RNA editing or exon skipping. So sometimes, say if your mutation is in an exon that isn't really necessary, and there have been a couple identified that may not be so necessary, then there are potential therapies that could skip over the one with the mutation. And so you're still going to have a neurofibromin protein that's different, or a Merlin protein that's different, but it might be better than it was before, right? And then we can also see at the protein level, we can have protein replacement, and so that's a big question, is how much neurofibromin or myelin do we even need, which is not that well understood at all. Myelin, we don't even know exactly what it does completely, so it's a challenge to know how much you would need to replace, but you can think if the problem is having a lack of protein, that introducing some protein that is normal might be a good thing. And then we can also have nonsense suppression. So some of those nonsense mutations can actually have the stop codon be ignored. And so then it will go and read through the way that it's supposed to. So all of these are potentials, right? And there's different types of therapy. All across this spectrum, they're being explored in NF and schwannomatosis. There's also something that we call suicide gene therapy, and suicide gene therapy is being looked at more so for those cancerous type of tumors, where a gene can actually be introduced into specifically tumor cells that will cause those tumor cells to self-destruct, right? And so if you're just really worried at the moment about dealing with this tumor, that's a potential. So how's it given? Who knows, right? That depends on the different types of gene therapy. And for NF and schwannomatosis, we have no idea what's going to come to market first, right? So some of these are going to be just given once and that's it. Some of them are going to need to be continuous or at regular intervals. Some of them might be something like IV, right? So you get an intravenous. And that might be something that, for example, impacts multiple tissues or a wide range of problems, whereas you might have something like an intratumor injection that is just trying to prevent cancer in that tumor, that's trying to prevent, say, a plexiform neurofibroma from turning into an MPNST, or treating something like a vestibular schwannoma to keep it from progressing. And then we could also have intracerebral injections, which are definitely the more scary looking ones up here, where they would have to insert the therapy directly into a brain tumor, for example. So some might be more invasive than others, right? Some might be once, some might be many, many times. So just to give you an idea of where things are in the NF space, these are just some updates from the 2024 NF conference in Brussels. So there were talks given on all of this type of work. So there's gene therapy. Oh, actually, that's my next slide. I was looking at the wrong one. This one is just about the status. So the gene replacement is actually in clinical work, not in NF, but in retinal disease, spinal muscular atrophy, there's an approved therapy for this, beta thalassemia as well. But the status in NF and schwannomatosis, we're just looking at in vivo models or in mice at the moment. Exon skipping, this is also referred to as antisense oligonucleotides. It's a long word for skipping the exons. This is also in a clinical phase in other conditions. So Duchenne muscular dystrophy has treatments that are using exon skipping. In NF and schwannomatosis, we just see this in cell cultures right now, so it's just in vitro. Nonsense suppression is also in clinical, also for Duchenne muscular dystrophy. In NF, we're still in cell culture. Protein replacement, also in that clinical space. There's several different diseases that are using protein replacement. Very difficult with NF and Schwann. And the NF1 protein neurofibromin in particular is very, very difficult to work with in a lab. We can't even get it to dissolve. And so to put things into solution, you have to be able to get it to dissolve. So there's a lot of challenges with our specific conditions. Gene editing, I just changed this on this slide because it used to say research, but there is now a approved CRISPR therapy for sickle cell. In NF and schwannomatosis, not really yet. And RNA editing and suicide gene therapy are just in research all the way across the board. We are seeing some cell culture in vitro in NF and SWN and in mice, and it's being explored pretty extensively in cancer treatment in general, right? But there's nothing that has come to market or in trials. So this is the slide with the meeting update. So just to give you an idea of what some of the stuff is that is happening in the NF world. Again, this is all very early. There's a lot of details on this slide that I'm not going to go through everything. But each of these was presented at the NF conference in Brussels. There's people actively working on them. So NF2 gene replacement, they're doing in mice at the moment and addressing vestibular schwannomas and glioblastoma as well. So there's different challenges to each of these with NF and schwannomatosis, and some of these are listed, but again, I'm not going to go through all the details on this slide because it's a lot. But you can see that we have NF2 gene replacement, NF1 exon skipping, so exon 17 is one of those exons that it seems like we could probably do without. And so if there's a mutation in exon 17 that causes a problem, why not just skip the whole thing if we can? Because it might be better off. Notice that some of these are temporary and some of them are permanent. So like with exon skipping, this is something you would need the treatment multiple times because it's only going to last for a certain period of time. And then you can see NF2 exon skipping as well. NF1 gene editing is happening a little bit in the lab, but this is just in cells, so we're just looking at NF1 impacted cells for this. Gene editing could potentially be a permanent solution, but like I said, that's something that we really are a long way from in the NF and schwannomatosis space. And then there's also some research going on in the NF and SWN community with suicide gene therapy, particularly with the other forms of schwannomatosis. This is in mice, and they've already started to observe from reverse pain behaviors from it, so it does look like it's having an impact on the tumors in mice. But again, this is just in mice at the moment. So it is something that is on the horizon, but we've got a bit of a ways to go. But as a community, we really are trying to understand patient needs and concerns. And so I wanted to share this study with you that I'm doing along with Kara Enstedt at NYU as part of the RANGE gene. You guys have heard a lot about RANGE, but as part of the RANGE gene therapy group, we've got a group that is really working, just trying to see how to make clinical trials the most effective when we get to the first ones, because we want to do it right? So this study is one of those discrete choice experiments that Vanessa was talking about. So it's used a lot in market research and basically gives you a whole bunch of options, a whole bunch of things for gene therapy A, a whole bunch of things for gene therapy B. You pick which one you would prefer. And then through the process of doing that a whole bunch of times, it kind of gets at the things that are most important, right? It allows you to weigh those risks and benefits. The thing that I've gotten from several people that have taken it already is that they actually learned some things about themselves when they were doing it, that they didn't really understand what the trade-off was. So as a part of this survey, too, there's a good bit of education, because education is important. So you learn a little bit along the way. For that reason, it is a little bit long. It's about 35 minutes long. It'll probably take you to take it. But I highly encourage you all to complete this, particularly those of you with NF2 and schwannomatosis. We've had an amazing response, which means that we're just going to be able to sub-analyze by more things that we didn't think we were going to be able to. And we really want to get a breakdown with NF2 and schwannomatosis. And so I encourage you, if you're impacted by one of those especially, to please fill out the survey, because patient opinions matter so much more than anything else, right? Because you guys are going to be the ones that are enrolling on trials when they do start. And that's all I have. So any questions, anyone? Oh, yeah.
Hi, could you give a layperson's understanding of mosaicism and how it might relate to NF2, and is that ever reflected on a genetic report?
Yeah, so mosaicism is a bit of a challenge all the way around. So for those of you that don't know, sometimes that germline mutation isn't really a germline mutation because it pops up a little bit later on in development, early enough to where, you know, there's some cells that are not going to be impacted, but a lot of other cells are going to be. So people that have mosaicism, it might not even show up on a genetic test originally because it might be testing a part of the body where they don't have BNF, right? So with mosaicism, very often it becomes important to test other types of tissues. And you know, with gene therapy, I can't say that there's anything actively going on. Like mosaicism would be a later step than the original ones probably, just than the full germline mutations, just because of the complexity of the different types of tissues. But yes, it can pop up on a genetic testing report if those cells are impacted. But sometimes it's going to take testing of other parts, like the cafe au lait spot, for example, right? Anyone else?
Any other questions? No? That's a heavy subject, gene therapy.
It is a heavy subject. It's a lot. Yeah, feel free to grab me anytime of the rest of the day and say, what the hell did that mean? Please explain it to me more. Because I am more than happy. It's a lot. This is a real challenge trying to educate people without any scientific background about gene therapy. But it's really important with this coming in the community that we all try to understand it a little bit better. Because the thing about gene therapy is this could potentially be something that is preventative. Right? This could be something that really changes the game for people with NF and schwannomatosis. It's just going to take a while to get there. So the more patient input along the way, the better.
It takes me back to years ago when we would be hearing about these drugs 15, 20 years ago, and you know, it comes faster than you think. So it is early in the game, but we want people to understand it, and there's a lot of questions about it too. So.
Yeah.